Good morning, everyone. Welcome to one another seminar for the fall seminar series. Today we have Dr. Nguyen with us from Johns Hopkins University. And before I actually give the floor to her, I would like to discuss some things about the graduate student community. So these are the people who have been contributing to make this possible all throughout the semester. Rodney and me are co-chairs for this year. We have Reza, Rajmohan, Tarun, and Rachel who are doing Facebook and email promotions all throughout the university. We have a support team of Anirban, Emmanuel, and Meghna who take care of the other things which are admin related work. And not to, I do not want to forget to mention Dr. Mistry, who has been a pillar for graduate student community, and he has been really helpful in making sure that the seminar series goes smoothly. Unfortunately we don't have him here today, but we really miss his presence. So GSC is an open forum. Anybody is free to join. We have weekly meetings at 4 p.m. on Fridays. Now we're doing it on Zoom. So anybody who wants to be a part of this graduate student community and want to contribute to the seminar series, they're more than welcome. Rodney and me, you could always approach Rodney and me. Here are our emails, and yeah, if you have anything for us, we would love to hear from you. So for OU students who are still not a part of the graduate student community, this QR code will directly take you to the Engage page where you could be a member. This helps us in showing participation to the Graduate Senate, and it helps us get funding and get to do more seminars like these, so it enables us more. So if you are a graduate student at OU or even an undergrad student at OU, and if you're not a part of the community, please use this QR code. And if you can't access this, then you can directly go to the Engage page using the link. I'll give 5 seconds for people who want to scan the code, and then I'll move on. Perfect. So this is our Facebook page. So any kind of seminar, so we update our Facebook page regarding the seminar that happened and the seminars which are coming up. So if you want to be updated on which seminar is coming up and if the topic is of your interest, you could always contact Reza. The email ID will be there in the event itself. You could email him for the Zoom details and you could join this meeting. So next Wednesday on 14th of October, we have Dr. Hantang Chin from Iowa State. And he's going to give a talk on 3D printing opportunities and collaboration. And then on Thursday, the next week on 22nd, we have Andrew Hortland, and he will be telling us his experience with the industry and some of the lessons that he has learned as his career throughout this time and in the industry itself. So today we have Dr. Nguyen, and I would hand it over to Dr. Lee. And Dr. Lee, please introduce and let us know more.
All right, thanks, Rashan. Today it's our pleasure to have Professor Vicky Nguyen from Johns Hopkins to give us our seminar presentation about her research. He's one of the leading experts in soft tissue biomechanics in our society. And just briefly mention about her biography. So Professor Nguyen received her bachelor degree from MIT in 1998 and her master and PhD degrees from Stanford in 2004, all in mechanical engineering department or discipline. So you will know how mechanical engineering, if our students, you will know how mechanical engineering can be really applied to different fields like polymer sciences or tissue biomechanics. Okay, so after that, she spent about 3 years in Sandia National Lab as postdoc scientist, and before she joined mechanical engineering department at Johns Hopkins. Okay, so now she's a full professor there and also the Marlin Zimmerman faculty scholar in the Department of Mechanical Engineering and Material Science at Hopkins. Her research centers around biomechanics of soft tissues and also mechanics of active polymers and biomaterials. Professor Nguyen has received so many awards in her early career, including 2008 Presidential Early Career Award for Scientists and Engineers, PCAST, the 2013 National Science Foundation Career Award, and 2016 Johns Hopkins Catalyst Award to restudy the micromechanisms of growth and remodeling of tissues. And if you're really working in applied mechanics, computational mechanics, you really know those big names. And Professor Nguyen really received a lot of awards because of her excellence in creative research. Just name a few from SME. So the inaugural HB Mechanics Award for Young Faculty in 2012, ASME Sia Nim and Nasser Early Career Award in 2013, and almost every year in 2015, she received Tom Hughes Young Investigator Award for Applied Mechanics. Now she is serving as an associate editor for the Journal of Biomechanical Engineering and is the president of the Society of Engineering Science. So without further ado, please join me to welcome Professor Nguyen for her seminar talk.
Thank you, Chung Hao. Do let me know if you're hearing me or if my internet is dropping. Sometimes that happens. As you know, we're in full-fledged Zoom mode here. So, you know, if there's any internet connection, let me know. I'll try and switch over to cellular for data, okay?
All right.
Let me share my screen. I really do want to thank Chung Hao and thank the GSC for the invitation to present and to meet you all and to present our, my lab's work. Can you see my screen? Can you see my screen?
Not yet, it's still loading.
It's still
It's coming slowly, sometimes.
Let me know when it comes up. Do you still have an issue not seeing it? Usually it's quite quick.
Yeah, you might try
maybe try,
slideshow.
yeah.
I'm sorry?
Try hitting run on the slideshow again.
Okay. So do you see the screen when I'm not running slideshow?
Yeah, we can see it now.
Okay. So let me try this.
Yep, it's good now.
Good. All right. Great. Pleasure to share our work, and I hope there's lots of questions. It makes this so much more interactive. You can interrupt me and ask questions, or you can wait until the end. Either way, I'm quite comfortable with both. It makes for a livelier seminar. And I do hope to speak to you after the seminar. One of the things that I do miss from giving talks at different departments is meeting people and meeting students in particular. All right, so the ocular system or the ocular tissues is a small field within biomechanics. So let me just spend some time introducing the eye and what its functions and the tissues are. So this is a cartoon of the eye. You can download it from NEI. And the eye is an optical system, and it's really a beautiful optical system. So here's a picture of it, and imagine that light enters the cornea, which is transparent. That's the transparent. It is refracted through the cornea, through the lens, and then the light signal travels to the retina back here. And that's where your photoreceptors are. Right? The cornea is responsible for 2/3 of your refractive power, 2/3. The lens is only responsible for the final third of the refractive power. And the lens are connected to these muscle fibers called signals. And what they do is they can tighten and flatten the lens. And that's how you can accommodate vision, meaning if you're squinting really hard to see something far off in the distance, what you're really doing is you're pulling on those zonio fibers and flattening the lens. And that changes your refractive power and allows you to see focus on near and far. If you're nearsighted, if everything works perfectly, then the focal plane is at the retina, which is supported by the sclera. The sclera is the white part of your eye. If you're myopic or far or nearsighted like I am, and most engineering students are nearsighted, then your sclera is a bit too long, or your eye is a bit too long, so your photoreceptors are behind. If you're farsighted, it means that your eye is a bit too short, so the photoreceptor is a bit in front of your focal plane. So the tissues of your eye have some very specialized function. The cornea has to be transparent, and the sclera and the cornea together form the eye wall, or the ocular coat, has to be rather stiff. Why does it have to be rather stiff? Well, for instance, your eye is pressurized, and your eye pressure varies throughout the day. It also varies when you stand up versus when you lie down. It varies a lot when you rub your eyes. If you ever rub your eyes really hard, you'll find that your vision's a bit blurred after, and then it adjusts. Now, if your eye wall is not stiff, imagine that throughout the day, your vision would be coming in and out of focus all the time, which is not something we want to do. You stand up, suddenly you become slightly farsighted, or you sit down or lie down, you suddenly become slightly nearsighted. That's not desirable. So your eye wall has to be rather stiff, and it has to be transparent, which is rather remarkable. Now, the photoreceptors at the end of, they're connected to a series of cells that end with the retinal ganglion cells, and the axons of those cells are very long. They run where they're connected to the photoreceptors back to the back of the eye, and they exit the eye through this small opening. This is called the optic nerve head, and this is an image of the optic nerve head here. And so you can trace each axon back to the location of the photoreceptor, and then you can associate each axon with its position on the visual field. So as I said, these tissues have to have some rather specialized functions. So they need to be stiff, they need to be tough and strong, because this is a delicate optical system that is exposed, right? So imagine through life, if you punch the face, you don't lose your vision mostly. That's because the ocular coat is quite strong. And the body is made out of very small number of proteins. One of those is collagen. That's the most common protein in the human body, in the body, period. And somehow the eye has to use collagen in such a way, or the collagen has to organize in such a way to make the cornea transparent, the sclera and the cornea stiff, and the tissues of the optic nerve head. Here's the lamina cribrosa, which is also collagen, supporting these axons as they exit the eye and into the optic nerve. When I first started working on ocular biomechanics, this was really my entry into biomechanics. I did not get my degree in in biomechanics at all. It was actually in fraction mechanics. But when I first started into biomechanics, I was really attracted to this picture right here of the cross-section of the corneal stroma. So the cornea is a multi-layered structure. It has a cellular structure called the endothelium, Bowman's membrane, Decimase membrane, Bowman's membrane, which are type 4 collagen membranes. All of them are very thin, and the thickest part of the cornea is the corneal stroma. That's the extracellular matrix material. And here's a TM, or transmission electron microscope, image of the stroma. The black dots you see are the collagen fibrils, and they're organized in layers called lamella. The fibrils are quite long in the cornea. They run from one end of the cornea to the other. In fact, they span more than just the length of the cornea. And they make this continuous, what I like to refer to as a fiber reinforced composite structure. Now I'm talking to a group of mechanical engineers and aerospace engineers, so you all should know what that means. And in fact, it looks quite a bit like a carbon fiber reinforced composite material for this TM image that you see here. what's really remarkable is that this is not an engineered material. It's a tissue. These are self-organized collagen materials. And the fibrils for the cornea to be transparent are uniform in diameter. Their spacing has to be within a certain range, and actually X-ray diffraction measurements or scattering measurements have shown that the spacing is really uniform. And that's what forms a transparent structure. The fact that these are fibers and there's 250 layers of these fibers also makes it a very strong and stiff material. Moving back from the cornea to the sclera, this here is the sclera. You can see that the sclera is also made out of collagen type 1, mainly type 1 and type 3 fibrils, also aligned mainly along in layers. But they're not as organized as in the cornea, and this is why your sclera is not transparent, opaque. Also, what you will see in the sclera is elastin, elastic fibers, another type of protein that behaves much more linearly with larger deformation than collagen. You will also see fibroblasts here in the sclera, much more than you will see keratocytes in the cornea. And the fibroblasts are quite active. And for this reason, your sclera will remodel through your life. It grows, makes your eye length grow or shrink, depending on how you use your eyes. Unlike the cornea's shape and dimensions are pretty much set after the age of 2. The sclera, on the other hand, can be used to remodel throughout your life, and mostly before an individual reaches maturity. In other words, if you have, it's a bit too late for you all, but for those with young children, make sure they don't always read a book for hours on straight, or watch video games, or what my kids do is stare at an iPhone a lot. We make sure that you look up, look at a distance for a long amount of time, and then before you resume near work. So excessive near work will cause myopia. And that's been shown in animal studies and also epidemiology studies. Now going back, and so this is where we've also spent a lot of time doing our work. And going back, here's the optic nerve head, is where we're spending the majority of our time right now. And I'm going to focus today my talk on our journey studying the mechanical properties of the sclera and the mechanical properties of the lamina cribrosa in the optic nerve head. So why are we interested in these tissues? The disease that I'm interested in is glaucoma. Glaucoma is the leading cause of blindness worldwide. Currently, it's estimated 80 million people have glaucoma worldwide, and that's estimated to reach 112 million people. It's a disease that is marked by progressive optic nerve damage at the lamina cribrosa, leading to loss of visual field. Okay, and the progressiveness occurs over a span of 10 to 20 years, depending on the acceleration of the disease. It's also associated with remodeling of the optic nerve tissues, and I'm going to talk more about this as we go along. It is a treatable disease. The only way to treat it is to lower eye pressure. However, it's not a perfect cure. Lowering eye pressure will reduce the progression of the disease in most people, but within the span of the disease, about 15% of people will lose the ability to read in one eye, and 5% will lose the ability to read in both eyes. And while it's not a disease that will cause, is not a morbid disease, meaning it does not cause mortality, it does impact the quality of life. And many patients, and I have family members who have been with glaucoma, and the fear of losing your eyesight is quite intense. Optic nerve damage is irreversible. Nerve damage is irreversible. So once the disease is detected and is diagnosed by loss of visual field at this point, which means that once it's diagnosed, vision's gone in those parts. So early detection, treatment, and adherence to treatment is absolutely important. And the focus of a lot of research these days is how to develop biomarkers for early detection. The earlier you can detect the disease, the more vision you can preserve. The number one risk factor, or the major risk factors of glaucoma, are intraocular pressure and age, and also race. Race and age are also risk factors. And here I have a plot of intraocular pressure and the numbers of persons within a population study broken out among normal, which means people who do not have glaucoma or a history of glaucoma, and those who do have a history of glaucoma. You can see that the population average is such that the intraocular pressure is higher in those on average with glaucoma than normals. So this is a very useful screening tool. Hopefully everybody here goes to see their ophthalmologist once a month, or an optometrist, once a year. And when you go to the optometrist for your yearly exam, one thing they'll do is they'll measure your eye pressure. This is when they numb your eye, and they'll either blow a puff air, or they'll use something called a Goldman applanation tonometer, which means they're actually pressing on your eye. And those instruments measure your eye pressure. For somebody who is young and you receive an eye pressure of say 21, which is the population average for glaucoma, most likely they'll tell you, come back, it's probably fine, come back and we'll measure your eye pressure again and we'll just monitor it to see how it goes. Now, if you're older, say 50s, then they will do more extensive tests to see whether or not you do have glaucoma or are at high risk for glaucoma. And that's when they'll prescribe treatments that will lower your intraocular pressure. For those who are African American, even if you're young and you receive a high IOP measurement, they will, optometrists and ophthalmologists will most likely request further tests because African Americans are at a higher risk for developing glaucoma. Asians, on the other hand, are also at high risk for developing a different type of glaucoma called angle closure glaucoma. But that is much clearer in diagnosis because the angles, which are the ocular outflow facilities for release of fluid from your eye, become blocked. And those can be detected much more easily than open angle glaucoma. In glaucoma, there is a pattern, typical pattern of vision loss. The axons, this is a picture of the optic nerve that's been stained, and where you have dark stains, that means those are viable axons. Where there are no stains, that means those are no longer axons, are no longer there. So the initial loss of vision is to the mid-peripheral vision. And this is one of the reasons why it's a really insidious disease. Is most likely you may not notice loss of peripheral vision. And it's not until that migrates further that into the central vision that you've lost a great deal of vision. So when you get a visual field test, and so most likely most of you have received a visual field test, they're not done here, but they are done periodically. You're up there with a light board and they're asking you to click when you see a light, and they're mapping your visual field there. And it's really hard to see those peripheral vision of visual. It's always one of the stressful tests for ophthalmologists to do. I mean, for people to do in an ophthalmologist's office, but it's really important because it maps your visual field. And also, as you can see from this, axons tend to be lost in the inferior and superior regions of the nerve. Dr. Quigley, Harry Quigley, who's my collaborator in 1982, did SEM imaging of the tissues. This is an ophthalmologist doing SEM imagings of the tissues. And he noticed that those regions tend to have greater porosity, so lower connective tissue density. So he hypothesized that regions of lower connective tissue density somehow have greater strains or stress. They did not have the language to discuss this, but that's what they're talking about. And that leads to loss of axons. Of course, they didn't have the tools to quantify it at the time. And also with the development of axon loss, you get this tremendous remodeling of the optic nerve head. Here's our two histological sections, and here's a TM image, again, taken by Harry Quigley in his lab. This black outline here shows the lamina cribrosa. And this is in a healthy, normal eye, and this is in an advanced glaucoma eye. You can see that as axons die, become damaged and die, there's this what's called an excavation of the optic nerve head. So it thins and it bows back, so to create this tremendous curvature. a detailed TM image of the stained elastin fibers of the lamina cribrosa shows that where the elastin fibers resemble straight taut fibers become really wavy in a highly glaucomic eye. And we still don't quite know what is the cause of this remodeling and how it contributes to the disease. So biomechanics, hopefully I've made the case that biomechanics is important in the development of glaucoma. This is how we view the role of biomechanics. You have mechanical loading on the eye. This is the intraocular pressure. That's the intracranial pressure that puts loading on the retrobulbar space to the eye. And then you have ocular muscles that are pulling on your eye. I mean, this is how you move your eyes. And so you can see left to right, up and down, things like that. All of those things exert a load on the optic nerve. Now, we do know that IOP is really important because if people who have glaucoma have higher IOP, tend to have higher IOP. If we increase IOP in animal models, they will develop glaucoma, meaning axonal damage right at the lamina cribrosa. This is what we see in patients. If we lower IOP in patients and in animal models, it does arrest the development of the disease. So we're going to focus on the loading of IOP, although there are people also working on to study the role of ICP in ocular muscles. So what does IOP do? Well, your eye is a pressure vessel. So those of you who take strength of materials, just remember PR/2T is the scleral stress that will be induced in the sclera by the intraocular pressure. In addition to that, you have the lamina cribrosa that's going to bend like a plate. So it's going to bow in response to an increase in the intraocular pressure. So pressure induces strains and stiffness, strain in the LC. And we believe that that strain or remodeling of the stiffness of the LC can activate astrocytes that can produce a neurotoxic response. It can perhaps injure retinal ganglion cell axons directly, or it can distort the blood vessels within the lamina cribrosa and cause an ischemic injury. All of these things actually, not just the astrocyte activation, but these things can cause remodeling of the sclera, remodeling of the lamina cribrosa, and it's an all big giant feedback loop that we're all trying to understand within the community. So some fundamental research questions that we've tackled, and this research I'm going to show you spans about 13 years, is what is the cause, or what are the strains, just some really fundamental questions. What are the strains caused by the intraocular pressure in the optic nerve head? What are the material properties of the tissues of the optic nerve head, and how are these altered by age and glaucoma? So really basic questions of how do tissue mechanical properties and biomechanical environment in the optic nerve contribute to the pathophysiology of the disease. And then next, once we learn that, we want to ask, does the optic nerve head biomechanics predict glaucoma risk, severity, progression? Meaning, if we can measure strains, can we associate strain with the risk or glaucoma damage severity or the progression of glaucoma damage? And then finally, we do want to get towards treatment. So the next step is to ask, does changing the biomechanics, does softening or stiffening the tissues of the optic nerve head slow progression of glaucoma damage? So I'm going to really discuss the first 4 bullets here. We have worked on this bullet here, and I'll talk to you at the end about the outcomes of that. But this is done in animal models, and today I'm just going to focus on the human work. So when I started at Hopkins and connected with my collaborator Harry Quigley, he made a provocative statement, which is nobody knows what the mechanical behavior of the sclera is. I'm like, oh, okay, interesting. No one knows how to test it. What we do is we just cut strips and we pull on it. I'm like, well, that's no good. Why is that no good? Well, because the sclera is, one, very heterogeneous in structure. It's got collagen fibers changing in various directions. So the anisotropy really depends on the position. It's also a curved tissue, and it's a really stiff curved tissue. So if you cut a strip out of the sclera and you flatten it, you really change the mechanical properties of the tissue. So the question is, you can do that, but really what are you measuring? So we developed to measure the mechanical behavior of the sclera, an inflation test. And we used something that was just coming to the community at that time, the biomechanics community, I should say, becoming in use, which is digital volume correlation. And because this is a digital image correlation. And because this is a curved surface, we use 3D digital image correlation, which is a misnomer because it's really a stereoscopic technique that gives you surface strains over a three-dimensional surface, okay? This is what I'm showing here is the setup in my lab. These are two cameras arranged in stereo. It's focusing on this inflation chamber here. And we received our specimens from donor human tissues from various eye banks. And they were shipped 24 hours post-mortem, and we would test them within 48 hours. That timeline means that my student had his cell phone on all the time, and whenever he received a call from the eye bank, they would ship it overnight on ice. He would come in the lab, pick it up, and we'd do the experiments as soon as possible. He would dissect all the tissues, intraocular tissues away from the sclera, clean out the conjunctiva. He measured the thickness using what's called an ultrasonic pachymeter. It's just an ultrasound probe that measures the thickness at different locations, and these black dots shows you the location. He would speckle with graphite, mounted into a holder that was designed to fit the contours of most eyes. We had 3 different sizes. Put it onto an inflation chamber. These are fiber optic lights coming in to illuminate that. You see this plastic line, that's a saline line. So we would inflate it using PBS. And it's connected to a syringe, which is connected or which is pushed down by an MTS machine. Why? Because we wanted to control both pressure and pressure rate. And the pressure was measured by the transducer, fed into the MTS machine. So we were basically using the controller for the MTS machine. And these cameras would image the deforming tissue, and we would use Big 3D to run digital image correlation to get contours or three-dimensional displacement fields. So we would get X, U, Z, and Y for the surface of the tissue. So what the program does is it reconstructs the geometry of the tissue and the reference geometry of the tissue, and also reconstructs the displacement field. And from that, we wrote our own strain algorithm to calculate things like circumferential strain and meridional strain. So circumferential strains would be the hoop strains that ring around the optic nerve head, and meridional strain would be the strains along the anterior posterior direction of the eye, so from cornea to optic nerve. A couple of things to notice right here. The black dot here is the optic nerve. It's a really compliant region. So I'm not going to show it because if I showed it, everything would be red in there and everything would be blue outside of it. So if you block that out, what you see is around the optic nerve head, high strains right around the optic nerve. Makes sense, right? Because you've got the optic nerve is a weak inclusion, is a soft, more compliant inclusion inside a stiff plate. So if any of you have taken elasticity, you can probably solve for this problem. This is a soft, kind of like a hole in a plate problem. It's more like a compliant inclusion in a hole problem. So you're gonna get strain concentration around there. The other thing to notice is that the meridional strain is a lot higher than the circumferential strain, and that's because of the collagen orientation. The collagen orientation is, the collagen fibers ring around the optic nerve head. They form, they're like ropes reinforcing that weak opening so that it makes it stiffer circumferentially, therefore smaller strain circumferentially than meridionally. My student Baptiste Coudrier had a really heroic stretch of experiments. He tested 55 eyes. Here we were plotting the results, stress, stretch, stress. This is the hoop stress, calculate using that PR over 2T equation for different age groups. So this is the most advanced age, and these are younger age people. Range between, we had one 26-year-old eye, but mostly in the 40s, 50s, and 40s and 50s. And you can see that there is a tremendous stiffening as you get older. This is not surprising. Everything in your body stiffens as you get older. It's rather, my age, it starts to get rather morose, discouraging. But yes, all your tissues get stiffer and less compliant as you age. We also did this comparing eyes in donors who were diagnosed with glaucoma and those who were not. And not only that, but we looked at, my collaborator evaluated the optic nerve that we were able to recover from these eyes, and based on his visual evaluation, was able to classify the nerves as damaged or undamaged. Undamaged meaning that there were very little detectable loss of axons in the optic nerve. And interestingly was that normal eyes in the peripapillary sclera, this region right here, closest to the optic nerve, were more compliant than those who had glaucoma, both damaged and undamaged. So it looks as if the sclera stiffens with age, but is the sclera of glaucoma eyes differ with glaucoma? So we, away from the peripapillary region, this is a region we call the mid-posterior sclera, we're able to use, again, Laplace's law to calculate circumferential stretch, and stress, the hoop stress, and we have DIC calculations of circumferential strain, so we can estimate what the stiffness is of that is, and we do see that it does age. It does not increase or change with glaucoma. The only place that changes with glaucoma is here in the peripapillary sclera. You can't use Laplace's law there to estimate stress because of the presence of the compliant inclusion. The other thing is that we're able to measure thickness, and we see that thickness does decrease with the donor age. We do see also that the degree of alignment decreases with age, and it's also different in glaucoma. And we measure fiber alignment using X-ray diffraction, scattering measurements, wide-angle X-ray scattering that was done by my collaborator out in the University of Cardiff. And we can, from these measurements, calculate the degree of fiber alignment, and we do see that they change with age. So we need to estimate, we know that the strain response is stiffer. We don't know if it's materially stiffer. So we need to be able to estimate the material properties of the sclera in that region. So we developed a method where we were able to use DIC to reconstruct the geometry of the undeformed configuration. So the configuration at baseline pressure, which was like 1 millimeters of mercury. We used the pachymeter measurements to reconstruct the thickness for each of the specimens we tested. We also had, because of our collaboration with the University of Cardiff, X-ray diffraction measurements, X-ray scattering measurements for every eye that we tested. So we were able to build a specimen-specific model for all of the tested eyes. The only region that we could not get good geometry measurements was right underneath the optic nerve head. There we used a generic model for how the optic nerve, how the sclera thins as it connects to the lamina cribrosa. Since we have this model, we were able to create it, and we have the strain, the DIC measurements for displacements along the surface. What we can do is then run what's called an inverse finite element analysis to predict what material properties are. Essentially, what that does is we run an FEA model, guessing at what material properties are to calculate the displacements, compare the displacements to the DIC measured displacements, and use that difference to predict whether or not we should increase or decrease the various material properties to get closer and closer to the measured displacement. So in other words, we're trying to minimize the error between the FEA and DIC displacements. And from that, we can then predict or work back out what the material properties should be given a certain constitutive model. So we used a pretty simple constitutive model. It's a hyperelastic model. It had 3 parameters. One, mu is for the stiffness of the isotropic part of the behavior of the material. Alpha is the stiffness, alpha beta times beta is the stiffness of the collagen phase, and beta is the strain at which the collagen material stiffens. And what do we see is that clearly there was a strong increase with age in these parameters. What you see here is a plot of the shear modulus mu for the isotropic component and alpha beta, which is the fiber stiffness. And it's broken out in two populations. One is the diabetic population. These are the black squares. And I mean, sorry, non-diabetic is the black square, and the diabetic is the white. Why? Well, because if you are diabetics tend to have greater collagen cross-linking because of the higher amount of sugars. So to talk colloquially. So we wanted to see what the effect was. And there was actually a rather strong effect. There's for both the diabetic and non-diabetic cohort, there was a strong stiffening with age, although there was a much more significant or higher rate of stiffening with age for the diabetic cohort. In terms of the fiber stiffness, the cohort with that was diabetic had a very stiffening with age for the collagen fibers. The ones that are not diabetic seem to have this increase with age, but it was non-significant. But that was an interesting result that we obtained. What about glaucoma? Glaucoma did not have as strong of an effect on the material properties of the sclera. We did see a general trend towards stiffening with the shear modulus and the fiber stiffness. However, the results were not significant. So we did not see as strong an effect on the sclera with on the material properties of the sclera with glaucoma. Although we did see a definite effect in the strain response. So the changes in, which basically means that changes in the structure was responsible for the increased stiffness, increased stiffer strain response of the sclera. We wanted to ask, how much do sclera properties affect the optic nerve head? One way to do that is we have the collagen fiber orientation measured by X-ray scattering for all of our tested specimens. So we can run a parameter study. What we did, and my student came up with the idea, is rather clever, is for every specimen, actually one representative a few representative specimens, and he would represent the collagen fiber structure as measured in the simulations. And then what he did was progressively made each point isotropic based on the threshold of the anisotropic ratio, meaning that if the collagen structure wasn't too aligned, then he just made it random. And he did this progressively, increasing that threshold at which he made things random. So you can see here, black is using the experimental measurements of collagen anisotropy, and white just means isotropic, a random distribution. So as with each run, he made the sclera more and more isotropic. To the point where he started affecting the collagen structure around the optic nerve head the most, because that's really where they were the most anisotropic. And he did this, and for each calculation, he calculated the difference with the physiological fiber structure, which means where we use the data, experimental data of collagen anisotropy for all of the elements. And he measured the difference in the posterior deformation of the lamina cribrosa. This is the posterior bowing or the posterior deflection, and the scleral canal expansion. So how much bigger, wider the lamina cribrosa get. And this red line here is when we started detecting noticeable difference. And this picture is associated with that red line. So we can really take away nearly all of the anisotropy, all of the structure that was measured, until you start reaching the region around the optic nerve head, which makes sense. Basically, that says that whatever collagen structure away from the optic nerve head wasn't really that anisotropic in the first place. It had a degree of anisotropy. Even if it had somewhat higher degree of anisotropy, mucking with those out there did not affect the deformation of the lamina cribrosa very much, the optic nerve head very much. It's not until you start altering the structure around the optic nerve head that you affect the deformation. But what was also interesting is, look at the numbers on the y-axis here. We could really alter the collagen structure, making the sclera more and more isotropic around the optic nerve head. And we had a significant effect on the scleral canal expansion. If you made the entire sclera isotropic, it would expand 50% more. That's what that 0.5 means. But it would only cause the lamina cribrosa to bow back by 0.5%. So not much. In other words, the posterior deflection of the lamina cribrosa, which could introduce quite a bit of strain into the tissues, neural tissues, were not that affected by the scleral properties. The scleral properties totally affected how much the lamina cribrosa expanded, so it's important in that way. But the other deformation with the posterior bowing was not too affected. So that was somewhat discouraging until we realized that imaging capabilities were catching up to us, and we can now measure the deformation of the optic nerve head. And that's what we spent the next few years doing. So this paper came out in 2017. We started working on this in 2014, which is how to measure the inflation response of the lamina cribrosa itself. This here is the eye, or the back of the eye. So before we were measuring the strains in the sclera. Now we're going to focus on that black hole right there. That is the lamina cribrosa, or the optic nerve head, or the lamina cribrosa is the connective tissue of the optic nerve head. How we're going to do it? Well, we're going to use a multiphoton microscope. So we designed a inflation chamber that can go into a multiphoton laser. And the objective is going to be placed right on top of this region so we can image and magnify and image the lamina cribrosa. And we do that in a 2x2 tile. So this image here that you see is a reconstruction of 4 images, so a 2x2 tile to reconstruct this. And this zoom is a 10x zoom. We're using a Zeiss LSM 710 LNO. We've now upgraded to an 810. We're imaging in backscattered second harmonic generation and 2-photon fluorescence. SHG is a technique in which you send in photons, 2 photons at one specific wavelength, and you get a photon back emitted at half the wavelength. And if you send in a photon at 790 nanometer in wavelength, and you get back one that's, 800, you get one back at 400, you know you've got collagen. So SHG is this really wonderful technique that we know is specifically imaging collagen. TPF is used to image elastin. We know what the excitation is and emission that we need to capture to image elastin. And we can do that in the same shot just because they're about the same. The excitation wavelength is the same. The image capture is different wavelengths. So we've got different filters for both, but we can capture them in the same experiment. So the imaging protocol is very similar to what before. We get the tissue, we prep it, we put it into the microscope. Because it takes quite a while to image, we have to make sure that we allow the tissues to creep. So we equilibrate for about 30 minutes, and then we image as fast as we can. And so we're only able to record quasi-equilibrium behavior of this tissue. And this is what we see. It looks terrible, right? So this is the lamina cribrosa as imaged by SHG. This is one plane of it. And then what we can do is that we developed a series of imaging processing techniques to bring out these features. So what you can barely see here after we run through deconvolution to reduce blur and local adaptive histogram equalization, you can see a lot more details. And with this, we're able to use the collagen structure and the elastin structure also as natural speckles to track deformation using digital volume correlation. Now, digital volume correlation is a cousin of DIC. It is a DIC technique, except it works in 3D. So you're tracking image voxels in 3D, so you'll get three-dimensional strain displacement field in a three-dimensional volume. And with that, we can do wonderful things like calculate displacements and strains in the optic nerve head, which, you know, when I first started, there was no way, no one thought that this was going to be possible. What I'm showing here are average through the thickness maps of the displacements in the X, Y, and Z direction, and the strains in radial, circumferential, shear directions, and then the maximum principal strain in the plane of the optic nerve, and the maximum shear strain in the plane of the optic nerve. And then here is a three-dimensional rendering of plot of UZ, the anterior posterior deformation. It's really cool, right? Because this is inflation. So with inflation, you expect somewhat this expected deformation pattern. But a lot of things became very clear. For example, there's this wonderful ring of compressive strain that develop around the lamina cribrosa at the junction with the sclera. You see local regions of high strains that develop. And so this allows us, because we get the full contour, this really does allow us to do a very detailed regional analysis of strains. And not only that, but because we're using the collagen structure as the natural speckles, we can also analyze the collagen structure to get its characteristics and understand why we develop the strain field that we do, right? So when we get the tissue, this is the contrast, after contrast enhancement. My student, Tracy, developed a series of steps, image processing step, that really brings out these beam-like structures that she can binarize and she can skeletonize. And once she develops the skeleton of that, she can do all sorts of fun things like measure the number of nodes and the node density. So the nodes are where these beams meet together. She can measure things like beam length, beam thickness, beam tortuosity or beam curvature, just to measure how wavy these things are. All of this will determine the mechanical behavior of this network. And so with the ability to measure strains and structure, we can look at how they change with age and glaucoma. And what we do see is that, again, we get a tremendous stiffening behavior with age. So with age, you get a decrease in the strains. We don't see network structure very much with age, and this tells us that the ECM material really does stiffen with age. And that happens through remodeling of the elastin collagen and for glycan structures, and also age-induced collagen cross-linking. We see that both structure and strain varies region. So here's the maximum principal strain. It is higher in the peripheral region than in the central region, and the pore area fraction is also higher in the peripheral than central. When we look at the inferior, superior, nasal, and temporal quadrants, we notice that the nasal quadrant, both peripherally and centrally, has a lower pore fraction, and it also has the lowest maximum principal strain, and these are statistically significant. And we see this not just with the pore area fraction, but with a variety of other structural measures, including node density, connectivity, and tortuosity. And what we see is that where we have regions that have higher strains, we have structures that correspond to higher strains that are then correspond also to regions that are typically more susceptible to axonal damage. So what we've done is we finally have quantified what my collaborator noticed in 1987, which 1987, so he was way ahead of his time, that the regions where he had lower connective tissue density was also the regions that had and are more susceptible to damage. And in fact, when we look at not just a global average or specimen average of these, but we looked at all the regional average of all the specimens and we put them together and we run a regression analysis, we see that all of these different relationships, that's really rich that we're going to be mining in future studies. Really interestingly, the most sensitive, the maximum principal strain was most sensitive to the pore area fraction. The maximum shear strain was the most sensitive to the connectivity. So how many of those beams came into contact at a node? this is so mechanical. It was just really a joy to see this result comes out. You know, if you're in structural engineering, this completely makes sense to you. We also saw a stiffer strain response in glaucoma. So this is a plot of normal eyes without a history of glaucoma, and these are all of our diagnosed glaucoma eyes. And we saw that there was a statistically significant stiffer strain response in diagnosed glaucoma eyes. When we break out the glaucoma eyes into those with mild damage and those with severe damage based on an evaluation of the optic nerve, we see really interestingly that it was the mildly damaged eyes that were stiffer than the normal eyes. But then when you go to the severe damaged eyes, you also saw a more compliant response than those of the mildly damaged eyes. And why is this? Why is this? It's because of remodeling. You start, as the tissue become more damaged, you see this, the lamina cribrosa thins and it bows out more. So that's, we think it's becoming structurally more compliant. But maybe we're not sure if it's becoming materially stiffer or also materially more compliant. But what we did see is that in addition to measuring strains and network structure, we measured eye length, the lamina cribrosa area, so how wide is the opening, and also the mean curvature. And we saw that eye length was greater, LC area was greater, and curvature was greater in glaucoma eyes. All of these should tell us that the region or the tissue becomes structurally more compliant. But we instead measured a stiffer strain response, which tells us that at least in the glaucoma mild eyes, the mildly glaucoma damaged eyes, the tissue is most likely materially stiffer. So the ECM is most likely stiffer in these eyes. Right, so let me move on because I'm running out of time. This leads us to the question. So we have measured strains ex vivo. Ex vivo means that the eyes have been taken out of their donors who've died. We removed the eyes from their support, biomechanical support, that's physiological. So the strain measurements we're getting is not physiological. It's a great way to get material properties and mechanical behavior of the tissue because we've isolated the tissue, but it's not an in vivo, it's not a physiological response. So can we measure strains in vivo? How is it different from that in ex vivo? And can we use the in vivo strain to predict glaucoma risk and progression? So how do you measure eye strains in vivo? Again, imaging technology caught up with us. And so we're going to use a technique called optical coherence tomography. This is an optical technique that is going to send infrared rays of light into your eyes. It's going to reflect them back. And the machine, I don't know exactly what the algorithms are, and this is the scary part of this part, is that we don't quite understand the optical black box. But what the images that we get are the images of the optic nerve head. All right, so we can image these eyes. And this here is a radio OCT imaging session. And we can do, for example, 24 of these scans forming 360 degrees to get the full field of the optic nerve head. So how do we change pressure? We can change pressure two ways. All glaucoma patients who have pretty severely high pressure or who choose to can get a surgery called trabeculectomy. So trabeculectomy is when they cut a flap in your sclera and allow the fluid to flow out of your eye to relieve pressure. So they cut the flap and then they suture the flap. And they put a drainage device in it and they suture the flap. And most of the time when they suture the flap, the eye pressure, because they do such a good job, spikes or is higher than what they want. So what's a very common post-op procedure is to have the patient come in and do what's called a laser suture lysis technique. That's when they just use a laser and cut away the sutures. And that allows fluid to flow out the eye and the eye to come back to normal pressure. And so we have a cohort of those patients. We've done 18 of those patients. They tend to be more advanced glaucoma. So to increase the, to also get people who have mild glaucoma and no glaucoma, my collaborator developed a technique where to raise eye pressure by wearing goggles. If you wear goggles incorrectly, and if they're too tight, they will press on your eye and they will raise your eye pressure. And so that's what we do. These are him wearing goggles where we remove the lenses so we can image through the goggles into your eyes. So the technique is this. If you're getting surgery, you get your surgery, and we measure your eye pressure before you go in to get your suture sliced. You come back out within the hour, we'll image your eye, measure your pressure again. For the goggles people, we'll do an imaging, measure their pressure, put the goggles on, wait 15 to 20 minutes, and then image, measure their pressures again, and then image their eyes again. And we'll do digital volume correlation of the before and after images, and we can calculate strains. And this is what we see. So this is the spectralis image, which is the spectralis is the machine. This is the OCT images. And you can see that it's, here's the optic nerve head. You can see some of the features. And then after we do our post-processing, you can see a lot more of those features. Those are the collagen beams or plates that are stacked up to form the optic nerve head. And again, we're going to use these features as natural speckles to do digital volume correlation. And based on these cleaned up images, we can also segment the tissues for a variety of reasons. One of them is when we calculate strains, we can average them within the tissues to calculate, for example, the average strains within the lamina cribrosa. This is the strain field, the EZZ strain field. EZZ is the anterior posterior position for a patient wearing goggles that elevate the pressure from 14 to 17 millimeters of mercury. You can see that that increase in pressure causes a compression of the lamina cribrosa. This is the strain field in one of the OCT slices for a lysis patient where the pressure was decreased by the surgical operation from 24 to 10 millimeters. You can see now large tensile strains that develop. Why tensile? Because when you relieve pressure, what was compressed becomes less compressed, and that is measured as a tensile strain. And we have 24 of these, so we can take the 24 slices and the 24 the strain measurements in those slices, and then reconstruct surfaces of these strains going down to the thickness. And this is, I'm showing here is the surface strains averaged through the thickness of the lamina. Just to show you that these are not random measurements, I'm showing here's EZZ, so strain, versus IOP change. And so if you have an IOP increase, meaning, you know, for our goggles patient, IOP increase, you get a compressive EZZ strain, and the IOP decrease for a laser suture lysis, you get the tensile strain, and you get a really nice linear regression. I'm going to skip over the ALD just because I'm running out of time. But to get to the chase, so what do we have? Well, in preliminary results, what we see is that there tends to be a trend in our goggles group. Remember, the goggles group are measuring really mildly damaged eyes and or undamaged controls, and we see an increase or decrease in compliance. So that's strain divided by IOP change with thinning retinal nerve fiber layer thickness. So a thinning nerve fiber layer thickness, meaning you're losing axons. So if you're losing axon, it means you're becoming more and more damaged. So we're getting a stiffer response with more damage. It's not statistically significant. We need more patients. In our lysis eyes, note where the retinal nerve fiber layer here is. We're getting the more severe people here, not severe people here. But in our really severe people, it's more compliant than our less severe people. This back is very reminiscent of our ex vivo measurements that show that the more severe people is more compliant than the less severe people because of structural remodeling of the optic nerve head. But the less severe people are stiffer than the no glaucoma people, which is what our goggles people are showing. So it's very promising, and we're going to push forward and hopefully get and definitely do more patients and hopefully get a statistically significant result. Progression. We also have some really interesting preliminary result that says that the patients who have a history of progression, meaning their visual field has gotten worse every year with every test, and those who don't have any progression, but those who have progression, they tend to have a stiffer strain response. This is super exciting, and we're going to, that's really awfully close to statistically significant, and so we're going to do more patients and hopefully show that this is true. Why is this cool? Because it doesn't take much to do these imaging measurements, and it does take a lot of computing power, but you know what, you just let the computer run. If you can use this as a biomarker, this is one more indication of an early screening process that will flag people for extra exams or for observation who may develop glaucoma in the future. So what conclusions thus far? This is an ongoing study. We have learned so far what the strain values are in these ONH tissues, what the material properties and how they behave. For the sclera, the material properties are still to be determined for the lamina cribrosa. We have strain measurements, but we don't have the modeling yet to get at material properties. We know how these are altered by age and glaucoma. They get stiffer with age. It looks like they get stiffer with glaucoma in the mild case, and the strain responses becomes more compliant with structural remodeling in glaucoma. Does ONH biomechanics predict glaucoma risk? Maybe. I think the early data looks very promising. Does stiffening and softening the tissue slow the progression? In our animal studies, which are mice, we do see that stiffening the sclera with glyceraldehyde, which is a cross-linking agent for collagen, worsens axonal loss. And when we use a pharmacological treatment that increases the compliance, we see a decrease in axonal loss. So again, this is a promising area of study for translational purposes. What's next? We need to do computational modeling to get at mechanical properties of the tissues of the optic nerve. We have strains, we have structure. It's now a matter of putting together that finite element model and running the inverse FEA. And then we also want to do the micromechanical modeling to determine, because mechanically it's really fascinating, what this rich network structure means to strains. And then also we're pushing into why does strain and high IOP or perhaps stiffness cause axonal damage, and we're doing that by studying the astrocytes in the mouse optic nerve head. So we're doing really lovely things here with imaging and strain measurements in mice which have really small optic nerves. So hopefully you'll hear more from that soon out of our group. And then with that, I'd like to acknowledge all the wonderful people who have worked on this project. It's, again, it spans going back to 2008. So it's really been a long journey, and it's very rewarding to continue pushing into the clinic with this, with our biomechanical measurements. And I want to thank you for being here and listening to the talk, and open up for questions.
Thank you, Vicky, for your beautiful talk and wonderful research. So we have time for questions. So maybe you just raise your hands on the participant window, and then we will pick your questions. So if you want to type your questions in the chat, we will facilitate that as well. So any questions from the audience? I do have a couple, but I want to. So Haley, the first one.
Hello, I did have a question. I hope it's not too far off topic. I was a little bit curious about your collaborator, Dr. Quigley.
Mm-hmm.
I guess I was wondering, it seemed like he was fairly largely involved in some studies involving scanning electron microscopy, and developing that goggles method for the pressure study. Would you consider his career path and participation in this engineering type research somewhat of an anomaly, or?
Chung Hao, I think we can both answer this together. I think, there's a pattern in biomechanics research that if you are an engineer and you find that medical collaborator who is going to be really excited, you form this really amazing team. And you see this a lot in just all fields of biomechanics. There's usually an engineer and there's usually a medical collaborator pair that will push the frontier. The reason being is that the medical collaborator really understands the specific of the tissue and the pathology in a way that we just, I'm developing the language for it, but man, the biology still's over my head. And we provide the tools that they need to prove a lot of these questions. So Harry and I, he's what, in his middle 70s now. He's been studying this area for a really long time. And he's just been an amazing resource in terms of bouncing ideas. And he'll, at this point, will bounce ideas off of me too. He'll come with, questions. And I'm like, well, yeah, we can measure that. So what we've done is, and it's so rewarding because what we've done is we've developed tools that then translate to his lab. So with the mouse work, I lead the human work, he leads the mouse work, but we developed the inflation test that they use that was developed by a former postdoc, Kristen Myers, in 2010. They still use that now. The imaging protocols, the DVC, we developed that for the mouse eyes in 2015. And we moved that over to their lab. They published a paper on it in 2017 using that technique. It's still being used. The imaging processing that we developed for the network analysis for the human eyes and for the mouse eyes, where we're analyzing astrocyte processes, we are now moving that to their lab for their people to use. So I don't think it's anomalous. I think you just have to find that right collaborator to work with. And when you do, it is a phenomenally rewarding process. Just because you can see the translation of your engineering work really quickly. I would never have gotten into the clinic without him. You know, he was the one who had that vision. I mean, not that he was the only one with that vision. A lot of people were going into the clinic and using OCT to image things like structure and how the distance between the lamina and the choroid changes. And so they can measure structure and they can measure relative position of structure. But it takes the engineer to go, yeah, you know, those displacements don't mean much. You really have to look at strains and how do you measure strains? And what do the strains mean? Strains are, you know, there's a tensor. So you have to kind of explain what the physical significance of all of those measurements mean. And it really takes an engineering thinking to do that.
Thank you.
Any other questions? Hey, Jin-san. Hey, Devin, you will be the next. Dr. Pei, it's your turn.
Okay, I just unmuted myself. Hi, Professor Wayne. Very nice to meet you here. I listened to you several days ago from that workshop at SES.
Yeah.
I really enjoyed what you said over there. That's why I decided to join this seminar, but I just finished teaching, therefore I'm on campus having to wear a mask. I wish to, really wish to learn more from you about multidisciplinary research. And, you know, I just, I told Chong Hao that I'd like to seek some professional advice from you in the future, you know, if, if possible.
Yeah, yeah. So I really like multidisciplinary research just because it allows you to work on bigger problems, right? So it does make for a lot more meetings, to be sure. But yeah, so I've been working with Harry for 12 years now. I have in my polymers world been working with Chris Yakaki when he was at MedShape, and then he became a professor at University of Colorado, Denver. For also just about as long. So in that partnership, he's the polymer person. He's the one who synthesizes all of these really interesting shape memory polymers and now liquid crystal and elastomers that we work on. You know, the advice there is to, you know, the way I met up with Chris is I went to a talk. I was interested in the area. I saw him talk. He was very engaging. I approached him afterwards. We talked, and he just had this willingness to listen to ideas, and I had a willingness to listen to his ideas. The same thing with Harry. When I got to Hopkins, I emailed just about every ophthalmology people that I could find on the directory. He answered my email, and we just, you know, started meeting together. I'm also a part of an EFRI on soft robotics, and that happened because my collaborator his student approached my student to do some simulations, and we just got, you know, we did that. It got published. We did a whole bunch of things on the side with him until we had enough preliminary data until we can run write a grant together. Those are few and far between. We're still trying to do that with other groups, and sometimes it works and sometimes it doesn't. So I think what's really important is just this effort, this effort to communicate, effort to contact, effort to do some extra work, effort to listen to each other. Not everybody does that. And when somebody does that, you don't really need to spend a lot of time with that person. But if you find somebody who's willing to listen to you, do listen back. And I guess my advice to the students here is, yes, it will be additional work. Your advisor may say, hey, let's meet with this professor. They've got some good ideas and they want to run some simulations. Would you be willing to do it? I would say, yeah, if you're not completely overwhelmed, try it. It may lead to something. It may lead for you to an extra paper at the very least. It may lead to a whole new direction that you had not even thought about. And so much of our work is serendipitous that way. I had somebody contact me the other day from the Department of Urology. They were working on some implants, some penile implants, and now they want me to be on a co-advise a PhD student, and so we'll meet about that tomorrow. So maybe that will work out in the long run, and it'll be fantastic. Maybe it won't. I had never even thought about working in urology, right? So I also was contacted from somebody who worked in gynecology. So we're doing uterine fibroid mechanical testing. Just keep an open mind, meet with people, spend some time.
Thank you very much.
It's cool. Let's see, Dr. Gan, I saw you unmute yourself. Looks like you also have questions. So
okay.
First.
Okay. And let me see if you can see my. Okay. All right. This is Ron Gan. It is a very nice talk and beautiful approach, and with all the experiment and modeling. Especially I like you focus on strength, because strength is mostly important parameter for measuring of the damage. And of course, strength is due to the stress, is due to the pressure. In your case, is mainly due to the it's called the intraocular pressure.
Mm-hmm.
I have a couple questions. First one is probably very basic, naive question, okay? Because you talk about the optic nerve head.
Mm-hmm.
Optic nerve head, it is a nerve tissue, or you are directly to determine the nerve tissue's mechanical properties, or it is combined together the different tissue. I a little bit, because I'm not in this area, so I just wanted to understand more the structure of these.
Sure. I think I have a slide for that. Let's see if I can find it. Apologize. If I'm not, I'll go back to the beginning and zoom in. Yeah, I don't have a slide for that here. All right, so let's just zoom into this one. So here's the, I could have sworn I had one. All right, so here's this, oh, you know what? Yeah, no, we'll do this. So here's the optic nerve head. All right, so this really bright ring, and what you're seeing here is the second harmonic generation imaging, SHG imaging. So anything you see is collagen. So here's this really bright ring of collagen fibers. That's the sclera.
Collagen fibers, yeah. Okay.
And then these are collagen beams. If you look in 3D, they're actually plates. They're plates, it's like a colander, plates with holes in it. And in the holes, this is now zoomed in of that. And this is a stained image that I'm showing from a paper in 1990. This is stained for something called GFAP, glial fibrillary acidic protein. So GFAB is a protein expressed by a type of cell called astrocytes. And astrocytes are the stellate cells. So let me go now down to here. I'm going to show you a mouse image right there. So this is a mouse optic nerve head, and what you're seeing here is stained astrocytes. A mouse optic nerve head looks just like a pore in the human lamina cribrosa. So all these network thingies, those are astrocyte projections. So they're glial cells that protect and physiologically support axons. Here's a zoomed in image, and in these black holes that you see, those are what we call axonal compartments. That's where the axons come through, and then the astrocyte processes would wrap around the axon. And then the other part of the astrocyte processes would reach out and connect out to the laminar beams. So the astrocytes form this really amazing network where they'll provide structural support. They also provide all of the ATP and signaling for the neurons. So the glia, microglia, blood vessels. So each of those beams also contains blood vessels. Those nutrient supplies have to come from somewhere, and collagen. So it's really a composite, very interesting composite.
So it is like a network, structure with the fiber, the collagen fiber, and the polar. Okay.
Yep.
And
it's
however, this is just.
Yeah, I was going to say it's hierarchical. It's just like all tissues in the body is. You have the collagen fibers forming a network, and then within the pores of those collagen fibers is also a network. It's a network of astrocyte processes, fiber and pore, and then the pore of those astrocyte processes are axons coming through.
Okay. So this is a typical the optical fiber, optical nerve structure. It is similar as the other nerve, structure like the optical nerve for the hearing. I just think about this. Okay, it is a similar structure for the nerve system, or just unique?
No, it's not. Yeah, no, it's not similar to the central nervous system at all. It's a very specialized structure. In fact, the astrocytes in the optic nerve are really different than the astrocytes in the central nervous system. They're always active. If you stain for GFAP in the central nervous system or the brain,
that's totally different.
It is very different. Phenotypically, it's very different. Yeah.
Okay.
The astrocytes in the optic nerve, they're always under stress. And the optic nerve is passing through a system that's always under stress, which is different than in your brain. Hopefully your brain's not under physical stress.
Okay, good. Okay. Yeah. Thank you. Could you go one slide? I like you have XYZ direction, also R theta direction, and then another one is the maximum, like you have once.
Like this?
This one number 19. Yes. Okay. So this one gives us for the displacement you use XYZ. Okay. For the strain, you use R theta.
Correct.
R theta Z is more in the shape direction. Okay, it is. And then you come down to the maximum. For maximum,
we calculate things in polar coordinates, the cylindrical coordinates. The tissue structure is, the optic disc is circular, and the circumferential alignment of collagen fibers is circular. So it's a more physiological, relevant coordinate system than a Cartesian.
Okay. Okay. It is still local plus the global together, or just from the different point of view. For displacement, you use XYZ, but for the strain, you use based on the shape. You use the R theta system. And then for the maximum, I'm a little bit confused by your maximum. Your maximum, E maximum, gamma maximum was UZ, okay? How to understand these, the
So
correlation?
Emax is the principal strain, the first principal strain in the XY plane.
Okay.
And
Okay.
gamma max is the maximum shear strain in the XY plane.
And the UZ is come to the Z direction.
That's right. Yeah, UZ here and UZ here is the same. It's just this one is a flattened image. This is a 3D.
Okay, good. Yeah, thank you.
Thank you.
Yep. So Vicky, I know we are running a little bit late for today's schedule. So do you mind taking one or two more quick
absolutely.
questions?
Yeah, no, not at all. I don't have anything in the next hour. So
I know they are raising their hand for a very long time, but anyway. So Devin, you want to go ahead to ask your question?
Sure. Thank you so much for the great talk. This is really fascinating to learn more about the eye because I've heard about it at some conferences, but it's really nice seeing a lot of the details. I just had a couple quick questions, if that's okay.
Yeah.
So for your in vivo studies, are you looking more at like the acute response to the pressure changes?
Correct.
Or do you expect that there's possibly something in the long term that's maybe affecting this?
Yes. So if my R01 gets funded,
okay.
Then what we're going to do is we're going to study the remodeling of the structure after pressure lowering.
Okay.
In some of these pressure lowering studies, you know, we'll get a few of these, but let me just show you. Go back to this plot. Yeah, this poor fellow had a intraocular pressure of, well, could be a woman, of 40, like 50. That's like pain threshold. So this poor person had a tremendous relief of pressure when that pressure declined. I would love to see what happens to that optic nerve after 6 months, a year and a half, 2 years. Just would love to be able to connect the remodeling to the strains that we measure at the initial outset.
Mm-hmm. Yeah, that's really fascinating. And on top of that, for the remodeling that you see, like I think you mentioned cross-linking or just remodeling of the collagen fiber structure.
Mm-hmm.
Do you have ideas of what the mechanism is for that? Is it cell-mediated or is it the astrocytes that you talked about?
Yeah, that's the reason why we're studying astrocytes, is because they not only nourish the tissues of the neural tissues, but they also are extracellular matrix producers and remodeling agents. Okay. There are a few cells in the optic nerve that do that. One of them is astrocyte. The other one is this weird cell called a lamina cribrosa that's somewhere between a fibroblast and a glial cell. So there's other groups studying that, but as you can see from so we think that those are major players. They also upregulate in terms of if you dump a bunch of TGF beta in them, they will produce MMPs. So we figured that they're a good target to study. Yeah,
that's sort of like the valvular interstitial cells that we have in the heart valve leaflets. Mm-hmm. I think they also respond pretty well to TGF beta. So I was just curious about that. Mm-hmm. But thank you so much.
You're welcome.
All right, I will probably pick the very last one. Marlin, you want to go ahead to ask your question?
Yeah, I just want to say also thank you very much. I really enjoyed the presentation, especially the different approaches and the experiments that were there. So you mentioned in the beginning, one of the first slides when you introduced the collagen beans, that you noticed that there was some wrinkling in the fibers with the patients. Yeah, and I think it was one of there was another SEM imaging, and you said that there was no specific findings to why those wrinklings happened.
Yeah. So I know what you're talking about, this one right here.
Yes.
Right?
Yeah. I just want to know if after doing all the strain values and seeing them in different locations inside the network structure, and especially that you said in some patients, mostly it was stiffer with some cases being compliant. Do you think there's a relationship between those wrinklings and the strain values that you found in different locations?
Yeah, I mean, it's indirect, right? We haven't made those connections. We haven't imaged down to this level yet, because these are TM images where you actually have to stain the elastin with a heavy metal to get it to visualize. But so I think what's happening is, and these are in very severely damaged people. What's happening is the tissue, the lamina cribrosa, goes from something that's relatively flat to something that's thin and completely bowed out like this. So, most likely that's the reason for the more compliant response with severe glaucoma. And that has to be connected to some disorganization of the laminar beams for it to be able to deform like that. We don't know exactly the nature of the beams and how they change with advanced glaucoma. We do see in our more advanced glaucoma patients, the curvature is higher. At the resolution that we're imaging right now with SHG, we can't see changes in the elastin structure like this. We can't see the waviness of the elastin structure. But we know from previous studies that elastin's turning over is changing during this remodeling process. And we know that there's collagen 4 being deposited into the pores. So there's other studies that tell us that there's extracellular matrix remodeling going on. But yeah, at this point, we haven't connected the strains to this level of remodeling at this length scale.
Okay, thank you.
Cool. That would be probably another multi-million-dollar project, right? Really study everything in detail. Anyway, so I think we are running out of time, and really appreciate Vicky for your time and to really give us this wonderful seminar talk. So just want to thank our speaker once again. So joining me to give a big applause to Professor Ran, and also as well as the follow-up dialogue, which is very nice. So I think that's pretty much for this webinar. And thanks again for everyone. And oh, by the way, Ronnie, do you have anything else for announcement?
I just wanted to say thank you also from me and from everyone at the GSC. It was an amazing presentation. Hopefully we can have you back again one day.
Yeah, I would enjoy that.
Sometime.
Yeah. Thank you so much, and have a good afternoon, everybody.
Yeah, thank you, Vicky.
Bye.
Bye.
Bye, Chung Hao. Thanks so much.
Yeah, thank you, Vicky.